Hi there, my name is Dr. Kreiss, and I'm here to tell you a bit about our work on label-free microscopy and deep learning for a better understanding of inflammation. Now, this work has been very interdisciplinary and has been carried out at different institutes, namely the University Hospital and the Faculty of Engineering in Erlangen, Germany, but also here at the Computational Optics Lab at Duke. Now, one great example of where inflammation is relevant are inflammatory bowel diseases. And in these groups of diseases, as the name suggests, there is a chronic inflammation of the bowel tissue. And there are many million patients worldwide with increasing prevalences among all ethnicity groups. If you want to know more about the current stage of IBD research, there is a good website I can recommend here. For now, all you need to know is that in the healthy case, your gut has a barrier of epithelial cells that protect the inside of your tissue against the outside of the tissue, where you have all the pathogens and bacteria and so on. Now, in some cases, there can be holes or breaches or small wounds in that barrier function, which lead to an influx of those pathogens into the tissue. That can even happen in the healthy case, but then your immune response would just fight off those pathogens, the wound would close, and that's that. However, in IBD, this immune response can be overregulated, which leads to collateral damage of the immune cells to that epithelial barrier, which then leads to more influx of pathogens, triggering the immune cells even stronger, which then causes this vicious cycle of ongoing inflammation. And that is really at the core of IBD. So as you can see, this process is very complex with many different components interacting with each other. And in order to understand and investigate certain aspects of the disease, people developed many different experimental colitis models to just investigate certain components. One example would be the so-called DSS colitis, where the damage to the epithelium is triggered first, which then leads to a destruction of that barrier function and then to an inflammation later. On the other hand, we have models like the so-called oxazolone colitis, where the immune response is really triggered, that then leads to a destruction of the epithelial barrier and then causes the inflammation, right? So we have something like epithelial-driven models or immune-driven models. Yeah, each of these models has a certain protocol, and there are also other models like chronic DSS, TNBS, transfer colitis, and yeah, in science, you always need control groups to compare these parts to healthy cases. So of course, there are also usually control animals involved. Now, if you want to look at the microscopic tissue structure of these models, currently the only way to go really is conventional histology. And in this process, you need to take out biopsy samples, embed them in the lab, slice them into very thin sections, then stain them by artificial dyes, and then finally doctors and pathologists would evaluate those images and give scoring grades of high or low inflammation. This process, as you can see, is very time-consuming and also very disruptive to the tissue itself. And this is where we really think label-free multiphoton imaging could be very beneficial to the process. Now, multiphoton imaging is based on the multiple photon or two-photon induced autofluorescence of certain molecules, where we can use a high density of infrared photons to induce, for instance, a two-photon absorption from the same fluorophore that you would conventionally excite with a single blue photon. Multiphoton imaging is also based on second harmonic generation, where the electrical field of the incoming laser interacts with the polarization properties in your sample to then induce higher harmonic signals, for instance, second harmonic. And these processes together can be used to derive molecular contrast from unstained tissue samples. So for instance, you can just look at the wavelength band of second harmonic generation from collagen fibers or from the autofluorescence signatures from certain molecules like NADH and FAD. And if you put all of this together, you get this multicolor image from an unstained, fresh, native tissue sample. So you just take out the biopsy, put it under this microscope, and you would see this type of optical histology. Now, this multiphoton process is naturally confocal, which means it is limited to the focal volume where we have a high density of photons. But that means we can just move the objective or shift the focal plane to image a different plane in the tissue without any autofocus light. It allows us to do 3D imaging to then use that data and put together these tomographic image stacks from a certain volume to look at the first epithelial barrier, for instance, and the deeper tissue layers that show us the mucosal crypts, immune cells, and all that interesting part that plays a role in IBD. And of course, we use that technology to look at all these different models that I've mentioned before. And already by naked eye, you can make out the differences between, for instance, the control case and the acute DSS colitis, but even between the other different models. So you can see that in each model, the tissue reacts differently. You can see different types of immune cells. You can see a different way of how that inflammation affects the tissue. And that really allows us to study these processes now with our label-free multiphoton signatures. The next step was for us to develop a label-free in vivo imaging system. So we built this device. It's a small, miniaturized, compact system that can be moved in and out of animal facilities, and it includes everything that a normal multiphoton microscope has, like the lasers, the amplifiers, photon multiplier tubes, all the electronics. And the key component here is a miniaturized grin objective that we use as an endoscopic head. And with that, we are able to do in vivo imaging in a living specimen without any surgery or invasive procedure. And we get the same label-free three-dimensional images just as before. Image quality is a bit lower because we have a lower numerical aperture of the system. But the great thing is we don't have to acquire biopsies to get this data now. Now, this label-free endomicroscope allows us to follow this progression of the disease over time. So basically different time points of this ongoing inflammation. So we can see from day 0 to the most severe day how the epithelial cells are destroyed. And we can run a very systematic study design where we keep taking biopsies from different animals at the same time points to then also compare the images respectively from the ex vivo imaging system. And since we had those biopsies, we also did the conventional H&E staining from those same biopsies. You can see that, the color representation and also the image quality differ between those three different modalities. But when you look at the underlying tissue structure that you see, you can see that they all agree very well with this ongoing destruction of epithelial cells and the increase of immune cells, and in some cases even scar formation at the latest time point. This is part of a nice video publication that you can check out here. So now let's bring this all together. We have a lot of data available for different animal models, ex vivo and in vivo, high image quality, low quality. So we have a bunch of data. And we also have certain annotations. For each of the stacks, we have our own multi-photon colitis score that we developed. For the respective biopsy, we also have the reference H&E score. And we have the experimental knowledge about which of the inflammation models it came from. So basically, we know if it was a DSS colitis or an acute colitis or a oxazolone colitis model. So all of these could be different types of annotations. And of course, if we have data and annotations, it's already very suggestive to do machine learning. And the goal here is to train a deep neural network with these label-free images as input to then derive the reference diagnosis. So let's look at the first part, the data. In this first run, I tested that approach with almost 200 image stacks, which is just a couple of megabytes per stack. When we look at the model, here I chose a SqueezeNet architecture, which is a very low capacity model that hopefully prevents overfitting in this data. I found a nice GitHub where there was a 3D conf version of this that allows us to leverage the 3D information about our approach. And that sums up to about 1.8 million trainable parameters. And looking at the annotations, there are different ways how we can slice this problem. In the first case, I looked at the multi-photon colitis score and interpreted it as a binary classification task just to find healthy and inflammation based on the image-specific annotation that we have. In a similar way, I used the H&E score just to define the reference H&E diagnosis of healthy versus colitis, giving the H&E score of the respective biopsy. And then finally, I think the most interesting pathway would be to do a multi-class classification problem where we use the label of the specific phenotype, which type of inflammation it was. And this leads to a 7-class multi-classification problem. So let me show you some of our preliminary current available results. For the H&E reference diagnosis, we can get a 5-fold cross-validation accuracy of 83%, which might seem low, but actually we found in our previous work that this is a similar level of agreement than different human annotators would have with each other. So actually it's not that bad. Very similar level of agreement for the multi-photon colitis score annotation. We are also getting close to 83% validation accuracy. And now for the multi-class problem, we get a 61% accuracy in the 5-fold cross-validation loop. This is pretty interesting because it shows us that, yes, it is indeed possible to distinguish different models based on the label-free input that we get from our molecular contrast imaging method. More interesting than just the mere automation, can the model learn, yes or no? I think it's also to go towards attention mechanism. So what I mean by that, if we remember from the beginning that each image in our stack is actually a 3-color molecular contrast image, where we have blue, for instance, collagen fibers, green epithelial cells, and red epithelial cells and immune cells, we can generate a weighted sum of those different channels in order to impose that weight as a trainable parameter to the model. So the model would then get that weighted sum as an input and not only learn to predict the task, yes or no, but also to optimize the weighted sum of that process. So in a sense, we can open that black box a little bit and understand which of the 3 colors, and therefore which of the 3 molecules, is most important. Here are some preliminary results. I show you the 2 binary problems first, where you can see that the model picks the green channels, the NADH, to be more important to derive that prediction. And that is very plausible because those show the epithelial cells. And the most apparent, the strongest change really happens in the destruction of epithelial cells during these models. So that's a plausible sanity check for us. Now, in the case of predicting the type of inflammation, the exact phenotype, this is a bit more complex, right? So there's not a clear winner under the 3 channels, which could be because it's still preliminary, of course, but it might also be because this problem is a bit more challenging and it's not just good enough to just look at the epithelial cells, but in order to make out the difference between different types of inflammation, the model then really needs to value all the different molecular channels and not just the epithelial cells. Where this approach would really be interesting is if we could train a model on the high quality ex vivo data, where we have full control, a controlled environment, high resolution, no motion artifacts, and a static clean environment, and we can just generate a bunch of data and train a really powerful model. But the biggest step would then to apply and deploy this model to the in vivo data of lower quality but much more clinical relevance. If we had this pre-trained model and if it would work successfully in the in vivo case, I think this would be a really powerful in vivo AI-driven inflammation detection tool. But there's still some work to be done, and this is just a concept for the future. For the current stage of this work, I really need to thank a lot of people who helped me to do this. As I mentioned, at the University Hospital and the Faculty of Engineering in Erlangen, but also here at the Computational Optics Lab. This project is currently funded by the European Union. If you want to know more about that, you can check out the website here. Yeah, thank you very much and hope to see you next time.